Physical and chemical aspects of the environment, such as temperature, light, moisture, soil type, and nutrition, have a marked influence on the nematode density, along with some biotic factors, such as the host plant agricultural practices followed. Because the environment can affect nematode pathogenicity through its effects on the host plant, prevention or avoidance of conditions which place the host under stress can offset nematode damage to crops. Agricultural practices such as irrigation, conservation of moisture by mulching, and using the correct fertilizer application are effective in limiting yield loss by increasing the plant's tolerance level. Such practices, however, only provide temporary relief as nematode population will rise under these favorable conditions. Nematodes increase to huge numbers without causing significant plant damage when placed in near optimal environment such as in a glass house. Now we will see the influence of surrounding environment and nematode. Many important relationships involving soil, physical and environmental factors and nematodes are established. Soil texture, soil moisture, structure and temperature are particularly significant. Soil characters. All plant parasitic nematodes complete part of their life cycle in the soil and are bound to be influenced by the soil environment. Nematode movement is directly affected by soil texture, soil structure through their effects on pore size. Soil physical and environmental factors and nematodes together are involved in several important relationships. Owing to the complex nature of soil, field distribution of nematodes are normally erratic and patchy. Nematodes are usually clustered, particularly among roots, and tend to migrate downwards as soil dries. Soil texture and structure. Pore size of soil have direct effect on nematode movement through the soil. Small pores in clay soil prevent nematode passage so that nematode must move in the spaces between aggregates. Even larger pores in coarse sands may be too big to allow nematodes to gain leverage between particles. This is the reason why nematodes tend to reproduce best in fine sands, sandy loam and well aggregated loams and clay loams. Soil temperature. Soil temperature keeps changing constantly and hence the greatest factors influencing nematode development. Diurnal fluctuation in temperature also vary to a great extent and depend upon soil type, texture, moisture, atmospheric conditions, latitude, elevation, season and soil cover. The temperature fluctuation are greatest at the soil surface and come down with increasing depth. The plant parasitic nematodes of the subtropical and temperate climates are most active during the warm summer months, when the soil temperature of 20 to 30 degrees Celsius are common. However, in tropical regions, nematodes multiply throughout the year. In tropical regions, nematodes multiply throughout the year, but in subtropical and temperate climates, most of the important plant parasites are most active during the warm summer months. When soil temperatures of 20 to 30 degrees Celsius are common, cool climate species often develop slowly in winter and become more active when temperature rise in spring. Water. Water acts as an important medium for active migration of nematodes in soil. Also, nematodes are dispersed passively by rivers, streams, irrigation canals, percolating water, and floodwaters. Nematodes normally live in moisture film surrounding soil particles and utilize the surface tension of this film for this movement. The film has to be neither too thick nor too thin. Too thick films may cause the nematode to slip, while thin films pose too much friction, which causes difficulty in nematode movement. Thus, there is probably an optimum soil texture and film thickness for each nematode species. Plant parasitic nematodes generally infect and develop best when the soil moisture is just below the field capacity. The spectrum of nematode population at a given point of time is a result of interactive ambient, biotic and abiotic factors. Biotic factors include temperature, moisture and aeration, texture, chemicals and pH. Host plants and microorganism falls under biotic factors. Meloldogyne characters. Members of the genus Meloldogyne are commonly called as root knot nematodes. Meloldogyne is of Greek origin and means apple-shaped female. They are an economically important Phalophacus group of highly adapted obligate plant parasites. They are distributed worldwide and parasitize nearly every species of higher plants. They reproduce and feed on plant roots. Root knot nematodes tend to induce small to large galls or root knots and result in necrosis of roots. The above ground symptoms are not readily recognizable, which may include yellowing of leaves, wilt. Now we will see about ecology. The frequency and density of root knot nematodes show dissimilarity from location to location due to variation in climatic conditions, soil type and agronomic techniques practiced for cultivation in each region. The rate of penetration depends upon the type of soil type and cropping system are probably the major components that play a vital role in root knot incidence. The density of plant parasitic nematodes also varies with each season. The predominant factors influencing the population of nematodes in soil include rainfall, soil temperature, soil moisture, rainfall pattern and host root growth. But the population of Meloldogyne incognita is mainly influenced by soil temperature than soil moisture. The temperature range between 26 to 27 degrees Celsius is favorable for nematode penetration. Also, the upper layers of soil are subjected to fluctuation with season than the deeper soil layers. Similarly, nematode population was more in summer and low in winter months. Because of the availability of fresh and actively growing roots, post-monsoon period is reported to be favorable for the survival and multiplication of root knot nematodes. Biology and life cycle of Meloldogyne. The life cycle begins from the egg, usually in single-celled stage deposited by the female. Development of the egg into an embryo starts within an hour of deposition, resulting in 2, 4, 8 cells, etc. There is slow movement of the embryo and the first stage larva within the egg. After the first molt, the second stage infective juvenile is formed within the egg. Hatching of larva occurs under suitable physical condition, but it is not dependent on host root exudates. The emerging second stage larvae are found free in the soil. The larvae which develop into female establishes feeding site in the pericycle region and becomes sedentary. Subsequently, 3 molts occur and the larvae develop into females. The neck region is unaltered. The larvae which later develops into an adult male is initially parasitic. After 3 successive molting, they leave the host in a worm-like manner and come closer to the females for copulation. Parthenogenesis is common in Meloldogyne. It requires nearly 30 days for development into a mature female. This might vary depending upon the species of the host and parasite and environmental factors like temperature and soil type. Heterodera. The first report of a cyst nematode attacking a tropical crop in a tropical area concerned sugarcane in the Hawaiian Islands. Heterodera is a genus of nematode in the family Heteroderidae. Nematodes belonging to the genus are obligatory parasites. Different species attack different crops, often causing great crop and yield loss. Nematodes of this genus are unique in that the females have the ability to transform into a tough brown cyst which protects the eggs. The name Heterodera refers to different skins of female and cyst. Ecology of this nematode. Species of cyst forming nematodes are less cosmopolitan but as pathogenic as root knot nematodes. The most widespread among them belong to Clobodera and Heterodera species. Heterodera avinae is a common species in temperate areas of India where it is the causal agent of a disease of wheat called molia. They cause considerable damage in the western countries of North Africa. The nematode is less restricted by soil temperature, persisting equally well in the hot Imperial Valley and the cooler coastal areas. It is also adapted to the complete range of soil types from the sandiest soil to the heavy clay soil to peat and muck soils. Poor sanitation practices and the movement of contaminated harvesting and cultivation equipment between regions have no doubt played a large part in the spread of this nematode. Biology and life cycle of this nematode. The life cycles of cyst nematode, that means Heterodera species, illustrate some of the possible modification of the typical life cycle. They are sedentary endoparasites. The second stage juvenile, that means J2, hatches from the egg and seeks a feeding site within the root. The juvenile 2 molts to the juvenile 3 and enlarges as the reproductive system develops. Nematode which develop into female or can no longer leave the root. They continue to grow as they go through the juvenile 3 and juvenile 4 stages. During this time, cells around the head of the nematode enlarge to form nurse cells or giant cells. Upon becoming adults, most of the eggs laid by just nematodes are maintained within the body of the female, which breaks through the root surface and is first visible as a white pinhead-sized spot or cyst on the root. After the death of the cyst female, the body becomes hard or tanned and protects the egg from adverse environmental conditions. Eggs within the cyst can remain viable for several years. Approximately 20 to 60% of the eggs hatch annually under suitable temperature and moisture conditions, resulting in new infection on susceptible crops. If host plants' roots are not available as soon as hatching, the juveniles tend to die. The optimum temperature for development and reproduction is 21 to 27 degrees Celsius. The length of the life cycle depends primarily on temperature and may be completed in 3 to 4 weeks at optimum temperature for reproduction. Because unhatched juveniles can remain viable for several years, this nematode is very persistent between host crops. In cyst nematodes, the adult males are vermiform. They are more common and are required for reproduction. It has been shown that males become more plentiful when food sources are limited. Tylenchulus nematode. This genus of nematode was first reported by J.R. Hodges in the year 1912 in California and described by N.A. Cobb in the year 1913. It is found in most citrus production areas and diverse soil texture worldwide and cause marked loss in citrus. Therefore, it is commonly called as citrus nematode. Citrus root nematode, the host range of this genus is very narrow. Now we will see the ecology and life cycle of this nematode. The citrus nematode is an obligate parasite reproducing only on living roots of host plants. It varies greatly with soil moisture, soil type and temperature. The number of nematodes in the soil as determined by soil analysis can give some indication about the damage potential of an infestation. Citrus nematodes exhibit sexual dimorphism at both the juvenile and adult stage. The life cycle is typical of plant parasitic nematodes beginning as an egg. Egg contains the first stage juvenile. Juvenile 1 molts within the egg to form juvenile 2 that hatches and searches for host roots. It becomes sedentary and establishes a permanent feeding site consisting of specialized cells called nurse cells, which are the main source of nutrients. The posterior portion of the female body swells with maturation and protrudes from the root surface, while its elongate neck and head ridge remains embedded into the cortex. Mature female produce eggs that are embedded in a gelatinous matrix secreted through the excretory pore. The length of the female life cycle from egg to egg ranges from 4 to 8 weeks. The development of the juvenile 2 male into adult is completed in 7 days and does not require feeding. However, the feeding apparatus, stylet and esophagus of adult males is poorly developed and may be difficult to observe. Nematodes of this genus are a sexually reproducing species that can occasionally reproduce by facultative parthenogenesis without the need of males. Rotylenchulus. Nematodes that fall under Rotylenchulus genus are semi-endoparasitic species in which the females penetrate the root cortex, establish a permanent feeding site in the still region of the root and become sedentary or immobile. The anterior portion of the body remains embedded in the root, whereas the posterior portion protrudes from the root surface and swells during maturation. Ecology and life cycle of this nematode. Both male and female nematodes can survive in air-dried soil kept at 20 to 25 degrees Celsius for 7 months. Local dissemination is through infested soil. Distribution is limited by low winter temperature, and nematode and disease development are both greater at 29.5 degrees Celsius than at 15 and 21.5 or 36 degrees Celsius. The adult males are non-parasitic, while the female is an obligate, sedentary, semi-endoparasites of roots. The species has an unusual life cycle. In spite of the presence of a well-developed stylet, the newly hatched second stage larva does not feed on and starves. After almost 7 to 8 days later, eggs are deposited in a gelatinous matrix outside the root tissue. When these eggs are placed in water, they promptly begin to catch. The life cycle is completed in about 25 days, provided the young females have found the host immediately. Rotylenchulus nematode as a semi-endoparasitic of sedentary nature induces a specialized nurse cell system for continuous food supply. Establishment of a multinucleate syncytium takes place. This involves expansion of cell walls of all the cells involved at the feeding site, wall dissolution followed by fusion of neighboring cells, protoplast, and finally formation of a multinucleate syncytium. These syncytia are mostly confined to the pericycle. Other pericycle cells are metabolically stimulated, but they remain discrete and uninucleate. The young pathogenic females destroy the exterior cortical cells of roots, and the damage increases when the nematode moves towards the phloem. Radopholus. Radopholus similis, the burrowing nematode, is the most economically important nematode parasite of banana in the world. Infection by burrowing nematode causes toppling disease of banana, yellows disease of pepper, and spreading decline of citrus. The burrowing nematode destroys the root tissue in the host plant. This leaves the plants with little to no support or ability to take up water and translocate nutrients. Due to the yield loss that it causes into citrus, ornamentals, and other agricultural industries worldwide, burrowing nematode is one of the most regulated nematode plant pests. Ecology. Burrowing nematode is native to Australia but is found worldwide in tropical and subtropical region of Africa, Asia, Australia, North and South America, many island regions. The widespread range of this nematode is due to its dissemination with propagative plant material. Especially infected banana comes, unlike most other plant parasitic nematodes, The impact of soil texture on burrowing nematode population levels varies with the host. Radopholus similis infection on citrus is favored by coarse sandy soil that is poor in organic matter, but is hindered by fine texture soil rich in organic matter. However, soil texture play a less important role on nematode population levels on banana. Life cycle of burrowing nematode. Burrowing nematode is an endoparasitic migratory nematode, meaning it completes its life cycle within root tissue. All motile juvenile stages and females can infect root tissues at any point along the length of the root. Males are non-parasitic and morphologically degenerate, that means without stylate. Penetration occurs mostly near the root tip. The cells around the site of penetration become brown. After gaining entry into the roots, the nematodes occupy intercellular position in the cortical parenchyma where they feed on the cytoplasm of nearby cells, causing cavities which integrate to form tunnels. After root penetration, these life stages mainly feed and migrate into the cortical parenchyma and also into the stele. The nematode completes its life cycle within 24 to 30 days at a temperature range of 21 to 32 degrees Celsius. Females lay eggs within infested tissues with an average of 4 to 5 eggs for 2 weeks that are produced through either sexual reproduction with males or by hermaphroditism. Eggs hatch after 8 to 10 days, and once the eggs hatch, the emergent second stage juvenile can migrate within the root and complete its entire life cycle within the root system. The juvenile stages are completed in 10 to 13 days. Buildup of the nematode population is favored by low soil temperature, adequate soil moisture, and availability of fresh tender roots. Plant parasitic nematodes are microscopic roundworms that must have living host tissue to feed upon in order to grow and reproduce. About 95% of plant parasitic nematodes live in the soil and feed in or on roots. The density of nematode population at a given point of time is the result of interactive ambient abiotic and biotic factors, which include temperature, moisture, texture, chemical nematicides used, pH, host plants, and interaction with other microorganisms. Biotic factors which impact nematode population in soil are many. Crop and cropping pattern influence the plant parasitic nematode population tremendously. Some nematodes have wide host ranges while others parasitize only selected hosts. Plant parasitic nematodes typically complete their life cycle in 30 to 60 days. However, some adults may survive for a year or two. The life cycle of a plant feeding nematode generally consists of an egg, 4 juvenile stages, and an adult. A typical nematode molts 4 times before becoming an adult. If females and males are both present, they mate and produce eggs. Females can produce from as many as a few to 500 or more eggs depending on the species.